Hello everyone, welcome to Biotechnotebook. In this video, we are going to see previous year GATE Immunology questions. First question is, dendritic cells are involved in cross-presentation of antigens. Which of the following protein or proteins is or required for cross-presentation? Okay, so the answer is basic leucine zipper ATF like transcription factor 3. Option A, P only. So the role of BATF3 is required for the development of dendritic cells. The dendritic cells are also known as antigen presenting cells. This is involved in cross presentation of antigen. Cross presentation of antigen is a process that allows our immune system to monitor viral infections or tumor or cancers. So in this process, it processes the exogenous antigens into peptides. Then these dendritic cells load these peptides onto MHC class 1 molecule. Okay, this MHC class 1 molecule initiates CD8 plus T cell responses, that is cytotoxic T cell responses against intracellular pathogen and tumors. Okay, so which is used to eliminate the tumor cells or cancer cells. Next question, which one of the following is required for the development of B cells in the bone marrow? Stromal cells are required for the B cell development in the bone marrow. Two reasons for that. One is it creates specialized niches in the bone marrow that supports B cell development, and also it produces some growth factors, okay, that stimulates the differentiation and proliferation of lymphocytes. Okay. Stromal cells create specialized niches in the bone marrow. This support the development of B cells. These stromal cells also produce some growth factors which stimulates the differentiation and proliferation of B cells. Okay. Next question is, which one of the following drugs is not an immune checkpoint inhibitor? Okay. So as we know that drugs are any substances other than food that is used to prevent, diagnose or treat or relieve symptoms of a disease or abnormal condition. Okay, this is a definition for drugs. So here we have to find in these four drugs which is not an immune checkpoint inhibitor. Okay, so the answer is option D, trastuzumab is not an immune checkpoint inhibitor. Okay, so these all four drugs are monoclonal antibodies, and this trastuzumab is a targeted cancer drug. This targets the HER2 protein, that is human epidermal growth factor receptor 2 protein, to treat cancers. This trastuzumab drug works by attaching to the HER2 protein on your cancer cells to stop them from growing and kills them. Okay, it works only if your cancer has HER2 positive. These three monoclonal antibodies are known as immune checkpoint inhibitor because it blocks the checkpoint proteins. These checkpoint proteins are generally found on the T cells and cancer cells. These checkpoint proteins doesn't allow T cells to kill cancer cells. So we have to block this checkpoint proteins. So we are using immune checkpoint inhibitors to block the checkpoint proteins. Thereby T cells can kill cancer cells. The next question is, match the immune tolerance mechanisms group 1 with the respective outcomes in group 2. So the first one is allergy. Allergy is a condition in which the body's immune system is failed to react to an antigen. Okay, so unresponsiveness to the antigen. So allergy means unresponsiveness to the antigens due to lack of co-stimulatory molecules. So P, answer is 3. Then activated induced cell death. So AICD is a process that causes the programmed death of T lymphocyte through apoptosis process. Okay, elimination of activated T cells after antigen clearance. Okay. So Q is 1. So next one is receptor editing. In receptor editing process, autoreactive B cell elimination takes place. Okay, so autoreactive B cell means B cells that react against the body's own substances. So it is a major mechanism in the bone marrow for maintaining B cell tolerance to the auto antigen. Okay, so it will eliminate autoreactive B cells. Okay, thereby it will protect our body healthy cells. The next one is regulatory T cells. Regulatory T cells is used to inhibit autoreactive T cells at periphery. Okay, so receptor editing process is used to eliminate autoreactive B cells. Regulatory T cells help to inhibit autoreactive T cells at periphery. Okay, it eliminate autoreactive T cells because both autoreactive T cells and autoreactive B cells react against the body's own cells or substances. So the two process mainly used to prevent the autoimmune diseases. So option 2 is correct. 3, 1, 3142. 3142. Option A is correct. Next question, match the cell type column 1 with its function column 2. Okay, so column 1 has some immune cells and column 2 has its function. How to match the cell types with its function? So first one is B cells. So B cells involved in humoral immunity. So B cells produce antibody specific to the antigen by humoral immunity process. Okay, next one is neutrophils. Neutrophils involved in phagocytosis. Phagocytosis is a process by which neutrophils engulf pathogens and cell debris to eliminate them. So neutrophils engulf pathogens and other cell debris to eliminate them by the process of phagocytosis. The next one is T cells. T cells involved in cytotoxicity. Okay, so in cytotoxicity process, T cells kills cells infected with virus. So T cells kills virus infected cells with the process of cytotoxicity. The next one is mast cells. Mast cells involved in histamine associated allergy. That means mast cells releases histamines and some proteases and cytokines into the blood, and it triggers the inflammatory responses. Okay, then this inflammatory response means it is an immune response that causes redness or swelling of an area on the body or tissues within the body. Okay. B cells involved in humoral immunity, neutrophils involved in phagocytosis, T cells involved in cytotoxicity, and mast cells involved in histamine associated allergic reactions. So 1, 4, 2, 3. You have to search for 1, 4, 2, 3. 1, 4, 2, 3. So answer B is correct. Okay, next question is CRISPR, that is CRISPR-Cas system is associated with. So the system is associated with option B, that is adaptive immunity in prokaryotes. Okay, let's see first what is innate immunity and what is adaptive immunity. Innate immunity is a body's natural defense system that you are born with. Okay, so naturally you have this immune system in your body and it is not specific. Okay, and it is the first line of defense system. So this is the system which protects our body from the pathogen once it enters into the body. Okay, next, adaptive immunity is body's learned immunity. So this immunity develops after exposure to particular disease, or you may get this immunity through vaccination. Okay, then it is very specific to a particular pathogen and it provides long lasting immunity. Okay, this is the difference between innate and adaptive immunity. Okay, now let's see how CRISPR-Cas system is associated with adaptive immunity in prokaryotes. So abbreviation of CRISPR-Cas9 is clustered regularly interspersed short palindromic repeats, and Cas9 is CRISPR-associated protein. So this CRISPR-Cas9 system is first identified in E. coli in 1987. So if bacteria is infected with virus, then this bacteria use this CRISPR system to capture and insert the small pieces of the viral DNA. So the virus which is infected this bacteria into their own DNA. Okay, so this CRISPR system will insert a small piece of viral DNA into their own DNA, so which allows the bacteria to remember this virus. Now it can quickly recognize and kill the same virus if it infects or encounters it again. So it provides a form of immunological memory which is similar to the adaptive immune system in higher organisms. Okay, so the system is known as adaptive immunity. Okay, so this CRISPR-Cas system is associated with adaptive immunity in prokaryotes. Next question is the process by which intracellular macromolecules are supplied for lysosomal degradation during nutrient starvation is. This is the definition for autophagy. So autophagy is the process by which intracellular macromolecules are supplied for lysosomal degradation during nutrient starvation. Option B is correct. Then apoptosis is nothing but programmed cell death. That is, it is a process that destroys cells that are no longer needed by the organism. Okay. So phagocytosis is the process of engulfing particulate matter, and pinocytosis is a process of engulfing extracellular fluids to clear the infections or to destroy the pathogens. Which of the following statements about immune responses are correct? Okay, so the answer is option A, P and R only. T cells are activated by antigen presenting cells. S T cells are activated by antigen presenting cells. Dendritic cells, macrophages and B cells are known as specialized antigen presenting cells, so which displays antigens to the T cell for its activation. Foreign peptides are not presented to helper T cells by class 2 MHC protein. So it's wrong. Foreign peptides are presented. Not presented is wrong. Foreign peptides are presented to helper T cells by class 2 MHC protein. Okay, so this option is wrong. So here they should give foreign peptides are presented to. Then next one is dendritic cells are referred to as professional antigen presenting cells. Yes, dendritic cells are referred to as professional antigen presenting cells, and macrophage and B cells are also known as professional antigen presenting cells. Okay, so option A is correct, P and R only. Match the autoimmune diseases in group 1 with the corresponding primarily affected organ in group 2. So group 1 has autoimmune diseases and group 2 has the organ which is affected by these diseases. So first one is Hashimoto's disease is an autoimmune disease which can cause hypothyroidism. Okay, hypothyroidism or underactive thyroid. Very rarely it may cause hyperthyroidism or overactive thyroid. So Hashimoto's disease will affect thyroid. Okay. The next one is juvenile diabetes disease. Okay, juvenile diabetes disease is a type 1 diabetes or insulin dependent diabetes. So in juvenile diabetes disease, the pancreas make little or no insulin. So this will affect pancreas. So next one is multiple sclerosis. Multiple sclerosis patient, the immune system attacks myelin in the central nervous system. Okay, so myelin is present in the brain white matter, and also it damages the nerve cell bodies which are present in brain's gray matter. Myasthenia gravis is a chronic neuromuscular disease that causes weakness in the voluntary muscles. So voluntary muscles include muscles that connect to a person's bones, muscles in the face, throat and diaphragm. So it affects skeletal muscles. So 4213. 4213. Option C is correct. Next question is match hypersensitivity types in group 1 with the corresponding conditions in group 2. First, let's know what is hypersensitivity reactions. Okay. Hypersensitivity reaction is an aggregated response by the immune system to a drug or other any substances. Adverse effect of hypersensitivity reaction may include symptoms like skin rash, swelling, difficulty in breathing, low blood pressure, rapid heart rate, nausea, vomiting, and dizziness. And sometimes it may cause severe life-threatening allergic reactions. Then there are 4 types of hypersensitivity reactions. Here they have given example for each type. So you have to match each type with its corresponding conditions. Example for type 1 is host reaction to B venom. Host reaction to B venom is considered as a type 1 IgE mediated hypersensitivity reaction. So in this type 1 hypersensitivity reaction, our body produces specific IgE antibodies against the venom protein. Okay, then this IgE antibody binds to mast cells and it causes them to release histamine and other inflammatory mediators, and it causes some allergic responses in our body. The allergic reactions include localized swelling and redness at the sting site, hives, itching, difficulty breathing, nausea, vomiting, dizziness or fainting. Then type 2 hypersensitivity reaction. So erythroblastosis fetalis is example for type 2 hypersensitivity reaction. This type 2 reaction is cytotoxic reaction, which one is mediated by IgG or IgM antibodies. In erythroblastosis fetalis, the mother's immune system produces IgG antibodies that crosses the placenta and it affects the fetus red blood cells. Okay, so mother's IgG crosses the placenta and it affects the fetus red blood cells. So thereby it leads to anemia in fetus. So it may be life-threatening without treatment. And next one is type 3 hypersensitivity reaction. This type 3 hypersensitivity reaction is mediated by immune complexes. So systemic lupus erythematosus is an example for type 3 hypersensitivity reaction. So in type 3 hypersensitivity reaction, there will be a formation of antigen antibody complexes. Okay, antigen antibody complexes which build up in tissues and it causes inflammation. Accumulation of immune complexes in tissues will lead to tissue damage and inflammation. Type 4 hypersensitivity reaction, it is also known as delayed reaction because unlike other types of hypersensitivity reaction, it takes several hours to days to respond to a particular antigen. Why? Because these reactions are mediated by T lymphocytes, that is cell mediated immune response rather than antibodies. So they take longer to develop. Allergic reaction become visible only 12 to 72 hours after exposure to the antigen. Tuberculin reaction is an example for type 4 hypersensitivity reaction. Okay, so this tuberculin reaction occurs when this tuberculin is injected into the skin of someone who has been infected with tuberculosis. So this tuberculin is purified protein derivatives which is used in a skin test to help diagnose tuberculosis. So it is used to detect the presence of the bacterium that causes tuberculosis, that is Mycobacterium tuberculosis. So the answer is 2134. So 2134, option D is correct. Type 1 hypersensitivity reaction is mediated by IgE antibodies, and type 2 hypersensitivity reaction is mediated by IgG or IgM antibodies, and in type 3 hypersensitivity reaction there will be a formation of immune complexes, that is antigen antibody complexes in tissues, and type 4 is a delayed reaction. It is mediated by some T cells. Next question. Which class of antibody is first made by developing B cells inside bone marrow? The answer is IgM. So IgM is an antibody which is first made by developing B cell inside the bone marrow. Next question. Which one of the following is not a part of the human non-specific defense system? The answer is antibody. Antibody comes under specific defense system. Okay, it is specific defense system. These three all comes under non-specific defense system. Okay, so specific defense system means antibody is very specifically react with antigen and it eliminate all the pathogens entering into your body. But these three are non-specific defense system. Why it is called as non-specific defense system means because it responds the same way to all the pathogens entering into the body. There is no specificity with this system. Okay, but antibodies or proteins which are very specific, but that is very specific to a specific antigen which is present on the pathogens. Next, let's see how interferon acts as a non-specific defense system. When a cell becomes infected with the virus, then it rapidly produces, that is, that cell, infected cell rapidly produces interferon. Then this interferon goes and binds with the receptor on neighboring cells and it triggers a production of antiviral proteins in neighboring cells which interfere with viral replication at multiple stages. And this interferon doesn't recognize specific viral strains. Instead of it triggers a general antiviral response in surrounding cells. So it comes under a non-specific defense system. Next one is mucus. It acts as a non-specific defense mechanism by creating a physical barrier. That is, the gel-like consistency of mucus forms a protective layer on mucosal surfaces like the nose, throat, lungs, and intestine, where it traps foreign particles including bacteria and viruses on its sticky surfaces, then thereby it prevents them from entering deeper into the body. Then saliva. Saliva contains various antimicrobial proteins like lysozyme, lactoferrin and mucins that directly inhibit the growth of bacteria and other microorganisms. Then salivary glands secretes IgA antibodies which can bind to pathogens and neutralize them. So saliva acts as a barrier against a wide range of potential invaders without targeting a specific pathogens. Next question. Antibody producing hybridoma cells are generated by the fusion of a. Okay, so hybridoma cells are nothing but hybrid cells. So here we are going to fuse two cells. So to make a hybridoma cells, you have to fuse B cell with myeloma cells. B cell has the property of antibody production and myeloma cell has the property of immortal cell growth. These myeloma cells are cancerous plasma cells that can divide indefinitely in culture, unlike normal B cells. That is, B cells have a very limited lifespan. So your hybrid cell has both the properties of producing antibody from the B cell and immortal cell growth property from the myeloma cell. Okay. So the resulting hybrid cells, also known as hybridomas, it can produce a large quantity of identical monoclonal antibodies that target specific antigen. Because immortal myeloma cells provide the ability to continuous division and proliferation, which allowing for the production of large quantity of antibodies. Specifically in this technique, we are producing monoclonal antibodies and the technique used is known as hybridoma technology. Next, let's see GATE Biotechnology Immunology syllabus. The first topic is innate and adaptive immunity, humoral and cell-mediated immunity, antibody structure and function, molecular basis of antibody diversity, T cell and B cell development, antigen antibody reaction, complement, then primary and secondary lymphoid organs, major histocompatibility complex, MHC molecules, antigen processing and presentation, polyclonal and monoclonal antibody, regulation of immune response, immune tolerance, hypersensitivity reactions, autoimmunity, graft versus host reaction, then immunization and vaccines. Then the books you may refer for your GATE preparation for immunology syllabus or Immunology by QB and Immunology by Wright. Thank you. Thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.